I'm so happy to be here to talk to you all about our work creating a single cell atlas of human and mouse-wide adipose tissue, but also more importantly, our work to try to make this a helpful and usable resource for everybody to explore gene expression in adipose tissue to help guide your projects. So I know that this is a NIDDK seminar, so maybe I don't have to answer this question, but one question that people may have is why adipose tissue? And I would say there's a lot of reasons why adipose tissue. For one, adipose tissue is found throughout the body. Sorry, let me just change this to a laser pointer. So adipose tissue is found throughout the body. You have visceral fat that tends to be more amongst the organs, such as epicardial fat, omental fat, retroperitoneal fat. You also have subcutaneous fat that's more right under the skin. These are not only notable for what's been studied about their differential contributions to disease state, but also because adipose tissue is everywhere, it's also communicating everywhere. It communicates with organs both in the traditional way that adipose tissue is thought of by liberating energy stores that can be used by organs that need energy stores such as muscle, but it can also secrete adipokines to regulate muscle in other ways, such as affecting muscle function during exercise. And muscle can similarly signal to adipose tissue. This is true about multiple organs throughout the body. Excuse me. Another thing that is interesting about adipose tissue is that it's a heterogeneous organ. One thing that you might think when looking at a slide of adipose tissue is that it's mostly adipocytes, because adipocytes are so large, they kind of overwhelm any other cell types that are in there. But multiple studies have shown that adipocytes only make up maybe about 50% of cell types in healthy adipose tissue. Other cell types in the adipose tissue include stem and progenitor cells, multiple kinds of immune cells, as well as multiple kinds of vascular cells. Finally, one thing that we know is that adipose tissue remodels during obesity. This is particularly important because we also know that there are a number of medical complications of obesity that may have something to do with adipose tissue. With adipose tissue, we know that during obesity, adipocytes become larger and more abundant, but also that they don't become more proportionally abundant, in fact, less so because of infiltration of immune cells. We know that there's large infiltration of immune cells, including pro-inflammatory macrophages, into the obese adipose tissue, and it's still unclear how these things are related, what it is about the obese adipose tissue that leads to the correlation of obesity with so many different diseases. So when single cell sequencing technologies were developed, to us, this was a perfect method to use to study adipose tissue. Like I said, adipose tissue is heterogeneous, and we know that cell type and cell type proportions change during obesity. So when you want to study obesity and you're looking at whole tissue, it's very difficult to tell where gene expression changes that you see are coming from. For example, are the gene expression changes due to the change of a gene within adipocytes themselves? Or are they due to the fact that there are now double the amount of macrophages that there were before? These aren't questions that can be answered with bulk RNA sequencing, but that can be answered with single cell RNA sequencing. So when these technologies were developed, we wanted to begin immediately. And so we used the technique that was first published, which was whole cell single cell RNA sequencing. In order to do this, we took fat biopsies from patients. We collagenase digested the fat, separating the mature adipocytes from the rest of the cells in the adipose tissue, collectively known as the stromal vascular fraction. Unfortunately, because, like I said earlier, adipocytes are so large, they didn't fit in the microfluidic devices that are used to perform single-cell RNA sequencing. So we were only able to sequence the rest of the cells, the SVF, in our initial experiments. However, we really were interested in adipocytes themselves. That were part of the reason why we wanted to do single cell on adipose tissue in the first place, was to see the changes in adipocyte gene expression in different metabolic states. So what we did is we moved to single nuclear RNA sequencing. For this, we were able to start with frozen tissue and do nuclear isolation, then load only the nuclei into the microfluidics device. As you can see, adipocytes, while they are large, their nuclei are normal size, or maybe slightly larger, but regardless, they were able to fit in the microfluidics device, and we were able to capture adipocytes as well as the other cell types in the adipose tissue. So we have two different cohorts that we ended up looking at. Our first whole cell cohort, we only collected subcutaneous fat, which is, again, the fat that's directly under the skin. We also only collected samples from female patients. As we were developing our single nuclear protocol, we also started recruiting from other surgeries where we were able to get not only subcutaneous adipose tissue, but also visceral adipose tissue, mostly either omental or preperitoneal fat, from the same patients during surgery. So for this cohort, we were able to recruit a larger BMI range, as you can see, as well as we included some male subjects in this data set, although this data is, again, mostly female. So what does the data look like? Here is the entire data set split by modality. So you can immediately see some differences between single nuclear and whole cell sequencing. One of them is just that we have a lot more cells from the single nuclear data set than we do from the whole cell data set. And that's due to having more subjects, more depots, as well as we just were able to get more cells, more nuclei sequenced because of various changes to the methods that we were using. But you can also see that there are some cell type specific differences. For example, there are these two populations of cells that you can see are present in the single nuclear data and pretty much not present in the whole cell data. As I mentioned, one of these populations are the adipocytes, which is the reason why we changed to the single nuclear sequencing in the first place. Another population is the mesothelium. This actually isn't so much to do with the change in modality, but because, like I said before, we were able to get visceral samples in our single nuclear data set. The mesothelial layer is a layer that forms on visceral organs, and so it's only present on the visceral, not the subcutaneous adipose tissue. Here I just have marker genes for these different populations that we identified in the adipose tissue, and I will get into subpopulations of these larger populations in a bit. So one thing that we're interested in, like I said, are the changes in cell populations as they're affected by depot and or BMI. So here I took this data and I split it both by depot as well as by BMI range. This is, I should say, only the single nuclear data to reduce bias caused by the modality. And this is also data that's been randomly subset. So each of these panels contain the same number of cells. So you can see that there are some obvious differences, especially by depot. For example, the subcutaneous fat, you can see even the adipocytes, even at this level, seem pretty sided by depot. Similarly, we can see that the ASPCs, or adipose stem and progenitor cells, are also quite sided by depot. We do also see some differences across BMI range. For example, the adipocytes, again, seem to migrate more to this end in the higher BMI ranges, and we see other differences in immune cells as well. So next, we also wanted to do mouse adipose tissue. Here for the mouse, we only did single nuclear sequencing, and we did both male and female mice, although more male, and in chow and high-fat diet. So here's the data split by chow versus high-fat diet. In this representation, it's a little difficult to see the changes that are induced by high-fat diet. But one really big change is actually an infiltration of macrophages, like I mentioned earlier, which you can kind of see here. It's the shape of the macrophages is less sparse on the right side plot, but we see a very large infiltration of macrophages in the high-fat diet, as well as other differences across cell types. Here again are marker genes for each of the cell types that we identified in this data set. And here also is the data split by depot. We don't see as strong changes by depot in the mice as we do in the human, but we still see some differences. For example, again, in the adipose stem and progenitor cell population, we see that there is some sidedness by depot. So one thing that we wanted to do is compare the human and the mouse populations at this kind of zoomed out level. Obviously, we, and I assume many of you, use mice as a model organism for humans. So we wanted to know how comparable mouse and human physiology are. And our overall answer is pretty comparable. You can see that for the most part, a mouse adipocyte, and I'll explain what this graph is showing a little bit in a moment, is similar to a human adipocyte. The way I did this analysis is I took all of the mouse cells and first plotted them by the cell type that they were annotated to in the analysis of the mouse data. I then mapped each mouse cell onto the human populations to see when integrated basically with the human populations, which of these clusters would each of the mouse cells fall into. And so here now we're looking at a river plot between the annotated mouse identity and the mapped human identity. And like I said, we see a lot of similarities between adipocytes, between ASPCs, between the mesothelium even. There are some differences here in the human data set. We had a small number of cells that we labeled as endometrial cells, which we think are due to endometriosis potentially in some of the subjects. Many of the subjects that we collected data from were undergoing gynecologic surgery. So we did see some weird results when looking at human populations that weren't present in the mouse data set and vice versa. But overall, we see at this level that there's a lot of similarity between groups. So next we wanted to look at these cell types more specifically. So what we did is we took each of these groups, we divided the data into adipocytes, adipose stem and progenitor cells, mesothelial cells, vascular cells, and immune cells. And we took each of these subsets and computationally separated them from the larger data set, and then reintegrated and recalled clusters to look at what differences we see between these cell types. And we do actually see a number of differences that in the case of the vascular cells, which are shown here, align with previous publications on vascular cell heterogeneity, not necessarily in adipose tissue, but in general. So in the vascular cells, we were able to identify stalk, venule, and arterial cells, as well as smooth muscle cells, pericytes, and lymphatic endothelial cells. We were able to identify them in the human and in the mouse. And we were able to identify similar marker genes for these populations in both. I did do mapping for all of these, but I just realized that I didn't put them in the presentation. So I'm sorry. But overall, what we see are that the mouse populations and the human populations at the vascular level map very closely to one another, suggesting that we're seeing very similar vascular cell types in both species. We next look at the mesothelial cells. Sorry, I should have cut this off. Again, we were able to identify different subpopulations of mesothelial cells. But as you can see here, while the overall marker of mesothelial cells, mesothelium, is similar, the markers for some of these specific mesothelial populations is different across species. So we saw overall less conformity between the human and the mouse subpopulations. For the immune cells, we found populations, both multiple populations of macrophages, monocytes, dendritic cells, multiple different types of dendritic cells, B cells, mast cells, T cells, and NK cells. Again, we found many of these populations in both mouse and human. There are some slight differences, but overall, as you can see, looking at the marker genes, we were able to find similar populations across species. Although looking at these plots, the top is the human, and the bottom is the mice, we can see that there's some difference proportionally between the mice and the human. The human proportionally had more T cells and NK cells, whereas the mice proportionally had more macrophages. This again is due to what I mentioned earlier, was the huge infiltration of macrophages that we saw in the mouse, where we didn't see something similar to that in the human. There are a number of possible reasons why that could have been. including the dynamics of weight gain between our cohorts. For the mice, we had just fed them high-fat diet. For the humans, they've probably been at that weight for many years. So, so the differences that we see here are largely in the number of cells as opposed to the types of cells that we were able to identify. Next, we looked at the stem and progenitor cells. Here, you can again see the human and the mouse. We were able to identify a number of populations that were similar, including ASPC1, ASPC2, and ASPC4, which maps to both of these. You can see also, however, that there are definitely some differences that we were able to see in the subclustering that didn't necessarily pop up at the whole cell level between these populations. Another thing that we wanted to do with the stem and progenitor cells was compare to other studies. Like I said, the initial studies that we did were only on the adipose SVF, which includes the stem and progenitor cells, as well as the vascular cells, and the immune cells, and the mesothelial, if you're looking at visceral adipose tissue. We were not the first people to publish whole cell, single cell sequencing on adipose tissue. So in fact, there was data from a few different groups that had looked at the stem and progenitor populations. So here we're able to compare populations across groups. And we can see overall, so basically, sorry, what I've done here is I've integrated our data with data from these previous papers, and then plotted each of these data sets by the cell type annotated by each author. So you can see that even though we're calling them different things, we are seeing very similar populations of these cells across species. Here, what we call an ASPC2 is similar to an ASC2, an FIP, a group 1, an FAP3, or a G1. And so this is really great to see that despite all of these different studies that are being performed in different countries even, that there's a lot of similarities across populations. So this is something that we want to look at further that I'll talk about a little bit more when I get to our future directions. Finally, we looked at the mouse and the human adipocytes. We were again able to subcluster these into different subpopulations with distinct marker genes. As you can see, again, though, these marker genes are not consistent across species. And in fact, the subpopulations that we find across species are fairly different from one another. So now I want to change gear a little bit and talk about how to explore the Adipose Atlas online. Like I said, one of the points of this paper was to create a resource for people studying fat tissue. And I should say we, after doing this analysis, spent a lot of time in the rest of the paper basically looking at these adipocyte subpopulations and trying to make educated guesses as to how relevant they are in different metabolic conditions. But for the purposes of this talk, I really want to talk about how this data can be used as a resource, and hopefully you can use it in your own projects. So one of the things that we did is we put the data on the Broad single cell portal. So I've blocked this with my little window on Zoom, but I hope that you all can see this QR code on the right, and it'll be on my last slide as well, if this is hard for you to get. But basically, if you just go to singlecells.broadinstitute.org and search adipose tissue, you can find it. And here we've uploaded all of our data sets. So I just wanted to walk you through it a bit so you could see the various ways you can use it online and also the ways that you can download it. So here we're looking at the human data. You can see on the right, you have options for what you view. Here you can select which data set you're looking at. You can look at the human data set, the mouse data set, and also all of the subcluster data sets that I just showed you. So here I've switched to the mouse fat. You can again see the mouse data sets here. And you can also, like I said, look at the subclusterings of all of the different vascular, immune, adipocyte and pre-adipocyte and mesothelium data sets. So one thing that you can see is that when you first switch to a subcluster, that this is still grouped by overall clusters. So here we can see the endothelial cells, the lymphatic endothelial cells, the smooth muscle cells, and the pericytes. So you can change what you're plotting this by when you look at annotation. So here, if you then select subcluster, you can now see this grouped by the subclusters that I just showed you that we annotated the data with. You can also plot gene expression on this data. So here I've plotted PROX1, which is a known lymphatic endothelial marker. And as you can see, it marks the lymphatic endothelial cells. And then you can even go back to the larger data set and look at PROX1 to see how good a marker it is for lymphatic endothelial cells across all of the cell types in the adipose tissue. You can also, if you look at these different tabs, plot the genes, plot genes using a violin plot. You can also look at UMAP plots at these feature plots for various genes. And for example, notice genes that are differential across pre-adipocyte subpopulations. So here I'm plotting PI16, which is more highly expressed in ASPC2 versus PPAR gamma, obviously a regulator of adipogenesis and a gene that's more highly expressed in pre-adipocytes that are farther along on the differentiation trajectory to become adipocytes, which we can see on the left. And so if you want to plot multiple genes and look at them across here we're looking at subcluster, but again, you can change the clustering and annotation that you're looking at. You can look at a dot plot of these genes and look at them across your ASPC populations or whichever populations you're most interested in. Next, I want to show you what's included on the summary page. If you follow the QR code, you'll go directly to the summary page. But if you just search for this, you'll go directly to the Explore page. But the summary page contains a lot of useful links that I would like to show you now. So one link is a link to the Seurat objects of all of the clusters and subclusters that I've just shown you. There is also this download link that's grayed out here, but won't be grayed out if you sign in, which you can do with any Gmail address. However, I think it's easier to download as a Seurat object, all of which can be found in this Google Drive link here. And here, if you scroll down on this page, there's a table for the download links for all of the human and the mouse data sets. This, I think, is really a good resource if you want to look at anything more than what I just showed you, basically. There are a couple more functionalities on the single cell portal, but there are differences as well. Or that you're unable to kind of look at some differences here. For example, if you wanted to split by modality, you can annotate by modality and look at the differences between them, but you can't look at, for example, both cell cluster and modality at the same time. So that's something that you would be able to do with the downloaded data sets, all of which are available to download at these links. Another thing that you can do is map your data to this data set using a tool called Azimuth. So Azimuth is a tool that's been developed, like I said, to map data sets to one another. We did not develop this tool, but our data is here as the human adipose reference. So what you can do is upload any single cell data that your lab has generated into this portal. And it will calculate, similar to what I showed between the mouse and the human, it will calculate which of these clusters or subclusters that we've identified your cells would fall into. So this can help with annotation, and it can also help with, like I said, harmonization of characterization of clusters and subclusters across data sets. So finally, I'd like to talk about the Human Cell Atlas Adipose Network. This is a initiative that I am co-leading along with Evan Roses, Suzanne Mondrup, and Ann Loft. If you're at all interested, please sign up for the Adipose Network. You'll see this QR code again. And so basically the Human Cell Atlas is, like the name would suggest, an initiative to create a consensus cell type atlas for a number of different human organs, including the adipose tissue, but also craniofacial, heart, brain, reproductive, liver, breast, pancreas, etc. So So we're obviously the Adipose network, and right now what we're really working to is grow the community of adipose researchers in this network. And this is to assist in a couple of initiatives that we're working on right now. One of them is to integrate data across labs. Like I mentioned before, one thing that we're really interested in and that we really think is important now is that now that we're identifying and consistently identifying in many cases some of these cell types across studies, the next step is to form a consensus annotation to look at all of these studies and create labels for these cell types that can be agreed on with the community. So one of these is to collect both human data and mouse data from any interested adipose researchers and integrate them together. For the human data, we're working with the Human Cell Atlas, which is also currently attempting to collect data sets from all of the different networks to create a large human cell atlas across Oregon. So they're currently leading the human data collection efforts, and then they will be integrating the data as part of the Human Cell Atlas, whereas we would like to take the adipose data and separately analyze that just amongst the adipose network. We also have not started collecting, but do plan to collect mouse data from interested groups. Again, collect from anywhere we can. This one won't be through the HCA, as there are many fewer privacy concerns when it comes to mice. And also we're going to integrate the mouse data across groups and then compare mouse and human adipose cell types. Another thing that we want to do is work on a white paper, which will cover some of the topics that we feel are most pressing for adipose and adipose single cell. So right now we've just worked on forming committees. I'm sorry, the committee survey already went out to everybody, so I'm giving this talk a little too late. But if you're extremely interested, please reach out and I'll get you in there. But basically, we're forming committees on cell type annotations, depot naming, and framework for reporting anatomical location. Like I mentioned previously, adipose tissue is all over the body, and we need to find a way to be very specific about where in the body we're collecting the adipose tissue samples from. Best practices for sample collection and processing, and best practices for bioinformatic analysis. The reason why I'm showing you this, despite the committees already being formed, is that the next step is an Adipose Network virtual meeting. We're planning this for early 2024. I think we've almost picked a date in early March. And so what we're hoping to do in this meeting is have presentations both on research from the network, but also from each of the committees about the progress they've made in the various topics, and as well as presentations from HCA representatives. Some of the other atlases, specifically the lung and the brain, are a bit farther along in their creation of atlases. So we'd really like to learn from them and what struggles they've had to go through in doing this large scale integration and consensus cell type annotation, so we can learn from their experience. And then, of course, we want to write a manuscript on the subject that hopefully will help make practices in adipose tissue, single cell and single nucleus more consistent. So like I said, I just wanted to show this again so you guys have a link to this Google form that you can easily fill out and sign up for the Adipose Network, so you can get information about the events that we've scheduled, including the meeting, like I said, that we're planning for early 2024. So a little bit more about our future plans. Like I said already, the first thing that we want to do is integrate human and mouse data sets to create a consensus atlas. This is, again, one of the things that we think is very important, is harmonizing the cell type annotation across groups. And so we hope to be able to do that both across different data sets that have already been published, but also across species, so we can be consistent and precise about what cell types that we're talking about. Another thing that we want to do is sequence other adipose depots, such as epicardial and bone marrow fat. to compare to existing data sets. We've called our work an Adipose Atlas, which I would say it is. But I'm sure some of you are working on fat depots that are not present in the data sets that we've collected already. So one of the next steps is to look at fat from other parts of the body to see how similar differences or difference they are to the fat that we've already looked at. And finally, we want to create an adipose tissue knowledge portal where users can explore existing adipose tissue omics data sets. Here on the right, you can see a screenshot that I've taken from the lipid droplet knowledge portal, which was created by Toby Walther and Bob Faris to look at lipid droplet data across here humans and flies and across multiple data sets. We want to build something similar for the adipose tissue. This is currently just a twinkle in our eye. We don't have this built yet at all, but this is really where we're trying to go, is create a even more comprehensive resource that not only is looking at adipose single cell and single nuclear data, but also bulk RNA sequencing data, proteomics data, metabolomics data, anything else that we can think of about adipose tissue. We really want to be able to collect this all in one place. So then when you query a gene, for example, you can look at it across species and across data type to hopefully get a better picture of what's going on with your genes of interest. So with that, I'm finished a little early, but I would like to acknowledge everybody who worked on this project, including Evan Rosen, my mentor, Lina Sai, the rest of the lab, all of our collaborators, the NIDDK. Like I said, much of the sequencing was paid for by this RC2 grant, without which I don't think we could have created an atlas like we were able to, as well as my funding. And here again are the QR codes, both to look at the single cell portal as well as to join the Adipose network. And I would be happy to take any questions.
Okay, thank you, Margo, for the great presentation. So now is our Q&A session. Feel free to type your question in the chat or unmute yourself to ask question. So I think in the chat is during your presentation is one message from I think it's Dr. Swapan. Das said, thank you for sharing your data, and they use your data in their recent paper. Yeah.
Oh, that's great. I'm so happy to hear whenever anybody tells me that they've already, looked at it on the portal or use it. I'm like, oh, that's great. Keep doing it, please. I'm so glad that you found it helpful.
Yeah. And then there's another question from the audience. I think it's during your presentation, I probably missed it. Could you show me how do I compare a pathway that I'm studying in mouse adipose tissue, whether to see whether it's present in a human adipose tissue?
Yeah, so what I would do is, depending on your familiarity with R, basically all of the analysis, I should say, has been done in R. And so all of the objects that are available to download are in Seurat, which is an R package. You can use this data in Python as well, of course, but I don't have it in this package process form as conveniently as I do for R. But you can download all the data matrices here. They're also on Geo, and build it yourself in Python, if that's what you're interested in. But for your question, how to look at a pathway that you're interested in, the easiest way would just be to look at it online. You can look at the genes from your pathway in the human data here. This is only looking at ASPCs, but you can change the clustering from human ASPCs to human WAT or any other human cell type that you're interested in, and then plot the genes that you're interested in on the data to see whether those same genes are showing up in the human data and whether those same genes are showing up are showing up in the cell types that you see in the mouse. One thing to note is that the human and the mouse gene names are different. Of course, the human gene names are all capital, whereas the mouse gene names are lowercase. And sometimes most of the time they're the same except for the case, but sometimes mouse and human have different gene names. So you do need to look up the human or the mouse genes that you've already identified and figure out what the human orthologs for those genes are, then plot those human orthologs on the human data. But you can do that either online or, like I said, you can download any of the objects, and it's a little bit easier to plot multiple genes at the same time on the objects if you're doing it in R. But you can also, like I said, look at as many genes as you want here on the portal.
Okay, and I think Dr. Swapan does raise a hand. You can unmute yourself now to ask question.
Yes, can I ask the question,
Of
Dr.
course.
Emont? Okay, so I have used this atlas, the data from this atlas and use it, but whatever that the current state, the number of samples are limited, and the phenotype data is very limited on these individuals, and the ethnic diversity is not there. These are all mostly European ancestry individuals. Do you think that you need to collect samples from who have been, physiologically phenotyped, more detailed, and then we can integrate that single cell gene expression data for better interpretation, and make it a more ethnic diversity, because there could be ethnic differences in the gene expression and the phenotypes. So just want to get your insight.
Yes, no, that's a great question. And so that's actually also one of the things that the Human Cell Atlas is really also finds important is the ethnic diversity. Like you said, most of our subjects were white, and we also didn't have a lot of metabolic phenotyping on those subjects. So we're right now working on some projects where we've been able to collect from a more diverse cohort of individuals, although it's still not there in terms of worldwide diversity, but we do have more Black subjects and more Hispanic subjects in our newer studies, but that's still, like you said, not nearly there in terms of diversity. We are also working on getting better phenotyping information, including things like we had BMI for this data set, but we didn't collect anything on insulin sensitivity or other parameters which are known to be more associated with the development of metabolic disease than BMI itself. So in our newer studies, we're collecting things like HOMA-IR, which is a measurement of insulin resistance. And we're hoping that that will make it a more robust data set. But like you said, it still isn't diverse enough. And that's where the Adipose Network comes in. We're really hoping to collect data from a number of groups around the world. We have we have members of the Human Cell Atlas Adipose Network who are in various countries, including Singapore. We know there's a group that's been collecting a lot of data. And so we're really hoping that by integrating our data with data from these other groups that are collecting around the world, we'll be able to expand the diversity, because like you said, there definitely can be things that we're missing when we're only looking at the cohort of surgeries that we're getting in Boston, basically, which is pretty limited.
Thank you very much. I actually have an African-American cohort, which is fully phenotyped for the insulin sensitivity by FSIVT and OGTT. And we have the genotype data, gene expression data, and the DNA methylation data and the metabolite data on all of these 256 individual African ancestry individuals. And I still have I have done the bulk RNA sequencing, but I still have the frozen samples. And I'm really looking for a collaboration for doing single cell analysis, at least for a subset of these people, so that we can understand the biological mechanism of insulin sensitivity and the obesity. So yeah, I'll be really interested to collaborate with you.
Yeah, please, like I said, well, I have the QR code here for signing up for the Adipose Network, but if you want to collaborate, please just send me an email. I just realized I don't have my email on any of these slides, but it's memont@bidmc.harvard.edu.
If someone can put that in the chat, that will be great. Thankful.
Okay.
Someone can put that in the chat.
Yeah, I'll do that after I finish presenting. But yes, definitely if you'd like to do something further than just share your already generated data, please reach out, and I'd love to talk about it.
Thank you.
Okay, thank you. And there's some question from chat. I think one comment from Dr. Ron Margo's great presentation on a valuable resource. And there's the next one, question from Dr. Neil McKenna. What insight have you generated regarding adipose cell-specific transcriptional regulatory networks?
So that's a great question. So right now, of course, the data that we've collected is all RNA data. But you can, of course, still think about transcriptional networks from that data. We've used packages like scenic, which is a package that was designed to look at gene sets in your different cell types and predict transcription factors based on motifs in the promoter regions of those genes. That analysis wasn't published because it was pretty unclear. So the next steps for that are a combination of doing more samples, but also of doing multi-ohm. 10x now, which we've been using 10x for our studies, has somewhat recently released multi-ohm kits where you can do both transcriptional and ATAC-seq profiling of nuclei in your single cell sample. And so right now we're working on optimizing that for adipose tissue so we can hopefully cluster these cells similarly to what we've shown already, but now also be able to look at the ATAC-seq data from those same cells to get a better idea of what's going on transcriptionally, doing motif analysis of the differential peaks, et cetera. So that's something that's currently ongoing. I know also we're not the first people to try the multi-ohm or the single nuclear ATAC-seq on adipose tissue, but so far at least we haven't seen any human data sets out there just yet. So we're working on that right now, both on all cell types, and we're trying to modify the protocol in various ways. I didn't go into this, but one thing that we've started doing is hashing our samples. So basically what that is combining your nuclear prep with the barcoded antibody, so you can combine multiple samples into the same run, thereby lowering the cost per run of each sample. So that's part of what's taking so long with the optimization, is that we're trying to add hashing to the multi-ohm paired SNUC ATAC-seq. So we're working on it, and I think that that's what's going to give us the best ideas of what's going on transcriptionally.
Okay, I hope that answers Neil's question. Let me move to the next question first. So next question is from Lindsay. Thank you. I've already used the portal and also download your dataset to look at a particular gene of interest. I'm coming across an issue where I'm wanting to look at statistical numbers beyond just a surrogate dot plot function. Suggestion on the best way to look at differential expression between different cell types in a human. My gene of interest isn't expressed in every cell, so I'm having issues with many zero values as well.
So if I understand correctly, you want to show the specificity of a gene to a specific cell type, and you want to perform statistical analysis on that. That's not something necessarily that I have done with this data. Is statistically look at the differences in gene expression across cell type. However, one technique that I have been using is called pseudobulk. With pseudobulk, what you do is you take your cell type of interest, for example, adipocytes, and here what I've been doing is splitting adipocytes, for example, into a low and high BMI range, and then in each sample summing all of the transcripts for all of, the reads for each transcript across cells. So instead of having, 100 adipocytes in your sample, you have one value of the summed expression for each gene in each sample. So you, like the name suggests, you're kind of recapitulating bulk RNA sequencing but you're only doing it on adipocytes in this case without the contamination of any other cell type. With the pseudo bulk, you're better powered to do something like differential expression analysis. So you can do differential expression analysis similarly to how you would do on bulk RNA sequencing data. So you can, calculate that, do a volcano plot, and crucially look at differentially regulated genes. So I haven't done this, but it should be easy enough to do the pseudo bulk and the differential expression analysis between two cell types. So you can, again, sum the counts for each sample for your adipocytes and for your pre-adipocytes, say, and perform the differential expression between adipocytes and pre-adipocytes, and then look at your gene of interest that way. That way you can calculate something like a p-value or an FDR. Hopefully that answers your question.